Greetings for the day, dear students. Today I'll cover a new topic, that is biological membranes and transport, and in this lecture I'll discuss about the sodium-potassium pump. Membranes. Plasma membrane is an envelope surrounding the cell, as we know. It separates and protects the cell from external hostile environment, and besides being a protective barrier, plasma membrane provides a connecting system between the cell and its environment. Now we'll see the chemical composition of plasma membranes. Membranes are composed of lipids, proteins and carbohydrates. Among the lipids, amphipathic lipids, that is containing both hydrophilic and hydrophobic compounds, namely PL, GL and cholesterol, are found in the animal membranes. Now we come to the structure of membranes. Lipid bilayer model, it was given by Devson and Daniels. Fluid mosaic model, it was given by Singer and Nicholson. FM model is more recent and acceptable. The thickness of the is 5 to 8 nanometer. A membrane is essentially composed of a lipid bilayer. The hydrophobic, that is non-polar region, face each other at the core of the bilayer, while the hydrophilic, that is polar regions, face outward. Globular proteins are irregularly embedded in the lipid bilayer. Membrane proteins. Membrane proteins are categorized into two groups. First is extrinsic, that is peripheral membrane proteins. Examples are cytochrome C of mitochondria. Second is intrinsic, that is integral membrane proteins. Examples are hormone receptors, that is cytochrome P69. The membrane is asymmetric due to the irregular distribution of proteins. Now we'll see the transport across membranes. The biological membranes are relatively impermeable. The membrane therefore forms a barrier for the free passage of compounds across it. Three distinct mechanisms have been identified for the transport of solutes, that is metabolites, through the membrane. First is passive diffusion, second is facilitated diffusion, and third is active transport. First we will see the passive diffusion, a simple process which depends on the concentration gradient of the particular substance across the membrane. Passage of water and gases through the membrane occurs by passive diffusion. The process does not require energy. The second is facilitated diffusion. This is somewhat comparable with the PD, since the solute moves along the concentration gradient higher to lower and no energy is needed. Facilitated diffusion occurs through the mediation of carrier or transport proteins. Specific carrier proteins for the transport of glucose, etc., have been isolated and characterized. Factors affecting the rate of diffusion. Number 1 is permeability of the cell membrane. Number 2 is temperature. Number 3 is concentration gradient, electrical gradient of the membrane across the substance across the cell membrane. Fourth is solubility of the substance. Fifth one is thickness of the cell membrane. Sixth is size of the molecules. Seventh is size of the ions, and last and the eighth is charge on the ions. Now the third one is active transport. Active transport occurs against a concentration gradient, and this is dependent on the supply of metabolites, that metabolic E, the ATP. Metabolic energy, that is ATP. This is also carrier mediated process like facilitated diffusion. Movement of substances against the chemical or electric or electrochemical gradient is called active transport. It is like swimming in the opposite direction of water flow in a river, also called uphill transport. The energy required is liberated mostly by the breakdown of high energy compounds like ATP. Active transport occurs with the help of carrier proteins, as in the case of facilitated mechanism is diffusion. Each CP can carry only one substance or more than one across the membrane. Those carrier proteins transporting only one substance are called unipods or unipod pumps. The carrier proteins carrying more than one substance are called sympods or antipods. Now we'll see the mechanism of active transport. When a substance to be transported across the cell membrane comes near the cell, it combines with the carrier protein of the membrane and a sub-pro complex is formed. Now this SP substance protein complex moves towards the inner surface of the membrane. Now the substance is released from the CPs and the same CP moves back to outer surface of the membrane to transport another molecule of the substance. Substances transported by active transport are both ionic form and non-ionic forms. Ionic forms may be sodium ion, potassium ions, calcium ions, hydrogen ions, chloride and iodide. As non-ionic forms are glucose, urea, etc. Types of active transport. Primary active transport. In this type of active transport, energy is liberated directly from the breakdown of ATP. By this method, sodium ion, potassium ion, calcium ions, hydrogen ions and chloride ions are transported across the membrane. Second is secondary active transport. When sodium ion is transported by a carrier protein, another substance is also transported by the same protein simultaneously, either in the same direction or in the opposite direction. This type of transport of a substance with sodium ions by means of a carrier protein is called secondary active transport. Carrier proteins, in this type, two different molecules in the same direction, that is sympod, and second is carrier protein transport two different molecules in opposite directions, that is antipode. Primary active transport of sodium and potassium ion, or this is called sodium-potassium pump. Sodium and potassium ions are transported across the membrane by means of common mechanism called sodium-potassium pump. This transport sodium ion form from inside the cell to outside and potassium ions from outside into the cell. This pump is present in all parts of the body. Cells have a high intracellular potassium ion concentration and low sodium ion concentration. This is essentially needed for the survival of the cells. High cellular potassium ions is needed for the optimal glycolysis and for protein biosynthesis. Further, sodium and potassium ion gradients across the membranes are needed for the transmission of nerve impulse. Carrier proteins of sodium-potassium pump. The carrier proteins involved in sodium-potassium pump has got 6 sites. 3 receptor sites for sodium ions. These are on the inner towards the cytoplasm surface of the protein molecule. 2 receptor sites for potassium ions. These are on the outer towards the extracellular fluid surface of the protein, and 1 site for enzyme ATPase, which is near the sites for sodium. ATPase consists of 2 alpha and 2 beta subunits and represented as alpha beta. Mechanism of action of sodium-potassium pump. 3 sodium ions from the cell get attached to the receptor sites of sodium ion on the inner surface of carrier protein. Two potassium ions outside the cell bind to the receptor sites of the potassium located on the outer surface of the carrier protein. The binding of sodium and potassium ions to carrier proteins immediately activates the ATPase, and ATPase causes the breakdown of ATP to ADP with the release of one high energy phosphate. Now the energy liberated causes some sort of conformational changes in the carrier protein. Because of this, the outer surface of carrier protein with potassium ions now faces the inner side of the cell, and the inner surface of the carrier protein with sodium ions faces the outer side of the cell. Now, dissociation and release of the ions takes place so that the sodium ions are released outside the cell and potassium ions are released inside the cell. The exact mechanisms involved in this dissociation and release of the ions are not yet known. Electrogenic activity of sodium-potassium pump. Sodium and potassium pump moves 3 sodium ions outside the cell and 2 potassium ions inside the cell. Thus, when the pump works once, there is net loss of one positively charged ion from the cell. The continuous activity of these sodium-potassium pump causes reduction in the number of positively charged ions inside the cell, leading to the development of a negative potential inside the cell. This is called electrogenic activity of sodium-potassium pump. This was all about the transport across the membranes. Thank you. Have a nice day.